I'd like to start by thanking Florence and Pierluigi for the invitation to speak. Of all the meetings I attend, this is by far the most rewarding, and I find it a huge honor to be asked to speak to you today. So as Florence mentioned, I kind of wear two hats at this meeting. I'm a presenter. I'm going to talk to you about my research, but I'm also a family member. My father died of CJD many years ago. So I have a personal connection and a professional connection to this disease. So as Florence mentioned, I've recently changed locations. I'm now at the Tan Center for Research in Neurodegenerative Diseases at the University of Toronto, just getting started with my own lab. But all the work that I'm going to talk to you about today was conducted while I was still a postdoctoral fellow in Stanley Prusner's lab at University of California, San Francisco. So why would one want to study prions using cultured cells, basically studying prion disease in a petri dish? So the most commonly used cell line for studying prions is called N2A cells. These are cells that are, these are mouse cells derived from a mouse that had a brain cancer. And then these cells can be infected with certain strains of prions, and then they become what is called scrapie N2A cells, or SCN2A cells, meaning that they're prion infected. And so the way you can differentiate an uninfected cell to a cell that is infected with prions is by looking for the biochemical signature of prion disease, which is protease or proteinase K PK resistant PRP. So normal N2A cells have none of it. Scrapie N2A cells have a lot of it. So this is a very simple system for studying prion disease. Why would you want to use a simple system? First, it's very easy to manipulate. Second, probably more importantly, as opposed to studying an animal that can take weeks or months or even years, you can get results in days with cells. And they're particularly useful for testing the efficacy or trying to find small molecules that might interfere with prion replication. So for instance, you can look for compounds that get rid of this protease-resistant PRP in the cells. So cells obviously are not perfect. They're not the best model for every situation. One of the things is obviously in the brain, when prions accumulate to a sufficient level, they're toxic and they cause the animal to die. That doesn't happen in cells, so there's a little bit of a discord there. And secondly, and most importantly, and this is what I'm going to focus on, is that only certain prion strains have ever been propagated in cells, so not everything can be transported into the cultured cell system. So just as one example of that, while I was at the Prusner lab, there was a large-scale effort to develop and uncover small molecules that interfered with prion replication. And so we conducted a large screen on using these SCN2A cells, which are mouse cells that are infected with mouse prions. And then we found an interesting series of compounds called the 2-aminothiazoles, called IND24 and 81 here. And when we tested these cells in mice infected with the same strain of mouse prions, we found that they significantly extended the lifespan of the mice, which was great. So this was essentially doubling the lifespan of prion-infected mice. So when you sort of extrapolate the lifespan of a mouse to that of a human, that could mean many years of survival extension. So this was very, very exciting, and we were very keen on this, and we thought, well, maybe we should actually try it on a strain of prions that's actually more relevant to a disease, so actual human CJD prions. And when we did those experiments, the results were very disappointing. So we found that when we treated mice that were infected, these are now transgenic mice, so these are mice that express the human prion protein instead of the mouse prion protein, and we inject them with little brain extracts from patients who had previously died of CJD. So two different strains, the sort of most common ones, so-called MM1 or VV2. And these are the survival curves of these mice. So red curves are the untreated mice. So these mice normally get disease at around 150 days of age. And when we treated the mice with the same compound that was effective against mouse prions, we found that they had no efficacy against human prions. So we concluded from that the efficacy of drugs is very much prion strain specific. And so the implication from that is quite straightforward, that if you want to develop a drug that's going to be effective against CJD, you need to study cultured cells that are replicating CJD prions. And this is where the problem comes, because this has never ever before been achieved. So if you take human neuroblastoma cells, sort of the equivalent of the N2A cells, but except human-derived cells, these have never been shown to be capable of replicating human prions. If you take the N2A cells and then put human PRP in them, it still doesn't work. If you take a cell line that I'm going to talk about called RK13, so these are cells that don't normally have any prion protein, but if you put in the prion protein from mouse, they become susceptible to mouse prions. If you put in the prion protein from deer, they become susceptible to deer prions. Fortunately, if you put in the prion protein of the human, they're not susceptible to human prions. And so this has been a major sort of bottleneck in the field, is that there is an urgent need to develop a cell line that can replicate CJD prions. And so maybe an alternative strategy is necessary. And so this is where my research comes in, for those of you who have seen me speak in previous years. So I'm the Bankful guy, so that's what I'm interested in. So in prion disease, normally, if you take a normal mouse and you inject it with prions from different species, so hamsters, humans, deer, sheep, you get very poor transmission. And this is because what's called the species barrier in prion disease, which basically restricts the transmission of prions from one species into another. And when I mean poor transmission, I mean that only a very small percentage of the animals that you inject will ever develop disease, and those that do develop disease take a long, long time to do so, 700, 800 days. So the complete opposite of that is the Bankfull. So the Bankfull is a rodent like a mouse that's found in Europe and in parts of Asia. And when you inject the Bankfull with prions from lots of different species, but in particular human prions, you get efficient transmission, meaning that 100% of the animals that you inject develop disease, and the incubation periods are fairly short. And this is all remarkable research that was done by the groups of Romolo Nono and Umberto Agrimi in Italy. So for various reasons, one being that Bankfulls are not endemic to North America, and therefore importing them over here creates a lot of regulatory problems. So we decided to try and make a mouse basically that mimics a Bankfull. And so what we did is we created genetically engineered mice, so-called transgenic mice, that instead of expressing or producing the mouse prion protein, they produced the prion protein from the Bankfull. And then when we took these mice and we challenged them with prions from all these different species, so mice, hamster, elk, sheep, cow, and 4 different strains of CJD prions, we found that they basically behave exactly like a Bankfull. So essentially 100% of the mice that we inoculated developed disease, and the incubation periods were relatively short. And you have to take relatively short with a little bit of a grain of salt, because obviously 394 days, it takes a long time, but in the prion disease field, that can sometimes be considered relatively short. And so more importantly is that typically when prions are going from one species to another, a lot of times you get the prions change. They sort of adapt to the new species. And if we want to have an accurate model of CJD, we don't want the prions to adapt when they're into these Bankfull mice. So we looked at the properties of the human CJD prions that had been passaged in these Bankfull mice to see, are they similar to the original prions? And we found that the answer was yes. So sort of the biochemical type, whether or not you have what are called type 1 or type 2 PRP scrapie, is maintained when you passage these prions in the mice. And the pathological signatures of the prion protein deposition in the brain are also very similar. So with CJD MM1, you get these little punctate brown stains of prion deposition, which is like the synaptic staining you see in CJD patients. With the VV2 strain, you get these more plaque-like depositions. And then with variant CJD, you actually get things that almost look like the florid plaques that are a signature of what you see in a patient with variant CJD. So we concluded that the property of CJD prions are maintained when you passage them in the Bankfull mice. So our hypothesis was then, if we could create cultured cells expressing Bankfull PRP, maybe that would allow us to study CJD prions in cultured cells. And so this is very much an ongoing research effort, and our preliminary results were quite disappointing. We found that we created these RK13 cells that express high levels of the Bankfull prion protein. We selected 2 clones number 4 and 37, and then we basically challenged them with lots of different prion strains that had already been adapted to the Bankfull sequence. And basically found that nothing worked. And so the most likely explanation for that is due to this sort of annoying problem that we get in prion disease research, is that certain clones of cells work and some others don't. And the reason behind this is not particularly clear, although research from Professor Collins's lab is beginning to illuminate why this might be the case. So research is still ongoing with this. I'm certainly pursuing this in my own lab using some different strategies as well as some new and hopefully better cell types. But all is not lost, so let me tell you about some of the other research that we've been doing that involve the prions in Bankfulls. So if you wanted to create a model of genetic prion disease in a mouse, the most logical thing to do is to introduce a mutation into, say, mouse prion protein that corresponds to CJD or fatal familial insomnia or GSS. And lots of people have tried this in the past, and the results have not always been very encouraging. So a lot of the time you get a disease that is quite atypical, that some of the mice don't ever even get sick, the disease isn't transmissible, and you don't get sort of like the classical signatures of prion disease. And secondly, if you wanted to try and create a model of sporadic CJD, in which case you have a normal non-mutant wild-type prion protein that converts into the disease form, then you would want to express wild-type PRP in the brain, and certainly this never leads to neurological disease in mice. So we were looking at our Bankfull mice, and so Bankfull prion protein comes in two different forms, much like human PRP comes in the M129 and V129 form. Bankfull prion protein comes in isoleucine 109 or methionine 109 versions. So the methionine 109 versions, they were normal and as they aged. Whereas the isoleucine 109 mice, we noticed that as the mice got older, they all started to develop signs of a spontaneous neurological illness that was consistent with the signs of prion disease that you see in a mice. And as we expressed more and more higher levels of prion protein in the brain, the faster the disease got. And this was a very, very reproducible illness. So 100% of the mice developed the disease. And so the role of this residue in prion disease is going to be explained more in depth by the next speaker, Dr. Castilla, who's done some amazing work on this topic. So we wanted to look, so this is a spontaneous disease. To what extent does it resemble real prion disease in a mouse? So here we're comparing mice that have been injected with prions to these mice, these Bankfull mice that are spontaneously sick. So just like real prion disease, these mice get the spongiform change, the vacuolations, the sort of Swiss cheese appearance of the brain that is very specific to prion disease. They have the deposition of misfolded prion protein in the brain, which is this brown staining right here. And they have a very pronounced activation of inflammatory cells in the brain, astrocytes, that are sort of a general response to neuronal damage in the brain. But of course, if we're going to call this a spontaneous prion disease, we need to show a few other characteristics of prion disease. And of course, everybody knows here that one of the big defining characteristics of the prion illnesses are that they are transmissible, i.e. they're infectious. So what we did is we took brain extracts from mice that got spontaneously sick and then injected them back into the same mice, except at a very young age. And when we did this, we found we got a very rapid disease incubation period, only 35 to 40 days. So in the prion field, this is very, very quick, basically the quickest disease that's ever been seen. And of course, because these mice develop spontaneous disease themselves, this is just showing that even when you inject the mice, that they come down at a much earlier age than they would if you just let them age spontaneously. And the last thing to see that it's a real prion disease is the presence of this misfolded protease-resistant prion protein. So initially we had problems finding this, but after some guidance from Romolo Nono, we looked a little harder, and then we were able to find it in the brain. So this is this little black band here. This is corresponding to spontaneously PK-resistant prion protein, and this is high concentrations of the protease, not in young mice, but present in sick mice, and then also present when we transmit the disease from the sick mice to the young mice. So basically, this spontaneous disease exhibits all of the hallmarks of authentic prion disease. And so we think that these mice are going to be a very useful tool for studying sort of the mechanisms that lead to the spontaneous misfolding of PRP that occurs during sporadic CJD. So now we've also taken this one step further, is because, well, now we have this Bankful PRP molecule, which seems to spontaneously misfold. What happens if then we introduce a mutation into it that corresponds to the genetic human prion diseases because, as I mentioned, attempts at doing this in the past have not been so successful. So we introduced 3 different mutations into Bankfull PRP. D178N, which is responsible for causing fatal familial insomnia, E200K, which causes familial forms of CJD, and then a mutation that chops off the membrane anchor of PRP, which is very similar to mutations that causes certain forms of GSS. And when we did this, there's a couple important things to note. So first of all of these mice developed spontaneous disease, so 100% of them. And the incubation periods weren't too long. But the most important thing to note is that we no longer required that we overproduce high levels of prion protein expression in the brain. So sort of physiological levels, the levels of PRP that would be present in all of our brains here, was sufficient to give 100% disease. So sort of 0.5 times normal level up to about 1 times normal level, or even very, very low cases in the case of the anchorless mutation. And I'm not going to stand here and say that these are perfect models of the corresponding human diseases. We still have a lot to work to do to fully characterize these mice. But certainly they produce different mutations produce different pathologies in the mice. So the anchorless one produced these huge amyloid deposits, which are basically present throughout the brain, much like you see in GSS. The E200K one gives these little speckles of PRP scrapie deposition, somewhat similar to the synaptic pattern you would see in E200K. And then the FFI, the D178N mice, have this sort of granular staining of prions. And so just to show you that basically the details here are not important, but the take-home message is that all of these diseases are infectious. So whether or not you infect them into young mice expressing the same mutation or into mice expressing the wild-type non-mutant Bankfull PRP, you get disease transmission. And like we saw before, it's very, very rapid, typically 35 to 45 days. So we hope that going forward that these mice are going to be very useful for studying these genetic human prion diseases. So in conclusion, I hope I can convince you that the Bankfull or the Bankfull prion protein sequence is a very interesting sequence to study because that these mice are highly susceptible to prions from all species, their tendency to adopt a misfolded infectious form on its own. And so we've concluded that Bankfull prion protein is inherently prone to misfolding and therefore is probably a good molecule for the basis of a lot of prion studies. So just in concluding, I'd like to thank especially Stan Prusner. All of this work was done in his lab with his support and the infrastructure he's created in San Francisco, as well as the guidance of Kurt Giles, all my colleagues at UCSF who contributed one way or another to these studies, especially for also to Romolo Nolo in Italy for the guidance. And of course, funding Canadian Institutes of Health Research and the National Institutes of Health. But most importantly, I'd like to thank the Creutzfeldt Jakob Disease Foundation and the donors who contributed to the research grant that funded part of the work that I've told you about today. So with that, I'll stop and thank you for your attention, and I'd be happy to take any questions.